dependent RNAs in an RNA-dependent fashion, so it's an RNA-guided RNA-binding protein. And when it binds to target RNAs, it activates a non-specific RNase activity that chews up all of the RNAs that are locally around this complex. So this is thought to be another type of phage defense system where the bacteria can recognize invading RNA genomes in an adaptive way that's dependent on the sgRNA. And if it recognizes those, rather than cleaving, rather than just cleaving in them, the bacteria, in a sense, commits suicide, where it just chews up all of its RNAs and the cell dies rather than allowing the phage to propagate. Jennifer and later Feng was able to show that this system can also be repurposed for biotechnology applications. So she showed that if you use an optimized fluorescent probe, you can detect specific cellular RNAs using C2C2 by a fluorescence assay. So what's sort of amazing is that this system can detect RNAs even down as low as like 10 picomolar in abundance, and this is borne out by this second paper as well. But I think this promises to be very useful for diagnostics in a lot of applications where you might be wanting to fluorescently visualize the presence of an infection within a biological sample, or the expression of specific RNAs associated with cancer progression, or you can imagine a lot of applications. Okay, so I'd like to end with one additional application of dCas9 that's been pretty popular over the last year or so, and some of this work was done by Mike. I'm not sure if you're going to actually talk about this, so just a little bit. I'll just say 2 words about it, and Mike can do the real justice to it. Essentially, there's been this desire to ask whether we can mutate the genome without inducing double-stranded breaks. So double-stranded breaks are a bit messy. They can induce translocations. We don't really have currently the ability to specify the DNA repair pathways that repair these double-stranded breaks. So the repair events, you can tune them between HDR and NHEJ, but they're still largely beyond our control. And so people started to ask, well, can we just use dCas9 fused to additional domains to mutate the genome in a specific way? And this is a process that happens normally during B-cell development and antibody maturation in all of us. And so what David Liu's lab and Mike Batzik's lab showed last year was that you could fuse dCas9 to these cytidine deaminases, either from rat or the human AID protein that's involved in antibody maturation, and then mutate the genome without forming double-stranded breaks. So these are targeted mutations that are induced in an RNA-guided way, and you predominantly, either depending on the fusion proteins you use, get C to T mutations, or in Mike's paper they showed that you can get C to N mutations. And so this is, I think, an additional application of dCas9 that people seem to be quite excited about, and there was a sort of an explosion of papers in Nature Biotech a couple weeks ago where people used one of these 2 systems to implement DNA editing without inducing double-stranded breaks in a variety of types of organisms, and showed a lot of applications. David Liu's initial work showed that you can also fuse on additional domains to increase your rate of editing. So they fused on a uracil DNA glycosylase inhibitor onto dCas9, and this increases the conversion of mutated base pairs, and that's detailed in the paper. I won't go through it for time, but both David and Mike have shown that this can be used to study anti-cancer drugs, or to, in theory, in a very specific way, mutate disease alleles from their pathogenic sequence back to a wild-type sequence, which could have therapeutic applications. And so I'll end there and just say I hope that I've convinced you that as awesome as Cas9 is, there is a place for thinking even beyond Cas9's canonical activity and using repurposed forms of CRISPR enzymes to do additional things in bacteria, yeast, and human cells. And we're pretty excited about that. So, thanks.
excited to discuss my favorite topic in the whole world, which is high-throughput CRISPR screens. And so I would encourage you guys to interrupt as often as you like to ask questions. I have a lot of material, but I'm happy to divert the flow to talk more about what you guys would be interested in. So Luke set up a lot of important topics that, in general, what we're going to try to do now is to take some of those technologies that have been engineered to work fantastically well in individual instances and now scale them up to do genome scale screens. And so first, I'm just going to give some general motivation for why one might want to do a genetic screen. And probably many of you have either done one or thought about doing one, but there's lots of reasons you might consider doing this type of thing. I'll describe partly by way of historical context, but partly by way of describing how exactly it is that the Cas9 system has been so powerful at the functional genomics scale, comparison to some RNAi technologies that were used early on in functional genomics. And in my own lab, we've done some direct comparison between RNAi and CRISPR that actually is somewhat illuminating in terms of understanding the difference between a knockdown phenotype and a knockout phenotype and what you can do with that information. I'll describe a number of different screens that have been done by a number of different labs. I'm going to focus some on the screens that we've done just because I can talk in more detail about them, but I'll describe some particular applications in terms of identifying the targets of different drugs and phenotypes for essential genes, which are very easy phenotypes to screen for. I'm going to describe a little bit a continuation of what Luke set up and using CRISPR-INA to do genome scale screens in really elegant work that's been led by Luke and Max and others in the Weissman lab. And throughout, I'm going to sort of try to give actually some tips and things that are sort of practical things that we've learned in the process of doing a lot of screens, as well as different design considerations that you might consider if you're thinking about setting up one of these screens. And then in the last bit, I'm going to describe two relatively new applications of CRISPR screening that we've been exploring in my lab, but have also been used in a number of other labs, and in particular, genetic interaction screens where you're knocking out pairs of genes to look at synergies between different pairs of genes, and then a scaling of the technology that Luke described at the very end of his talk, which is what can you do if you can mutagenize a large stretch of DNA. And then at the end, I'll just describe some useful things for what do you do when you have a screen, the results from a screen, how can you actually use that information. So just one really basic slide, I think, to kind of understand where screens have come from. Probably this may be too basic for some of you, but before the sequencing of the genome in the olden days, and actually still today to some extent, the way you would do screens would be using forward genetics. And here you take an organism, you mutagenize it, you select for some kind of a phenotype that you're interested in, say resistance to a drug, and then you find the gene that's responsible for that phenotype, usually by complementation or something like that. And this was, for a very long time, the way that people did genetics in an unbiased fashion. Of course, you can also study heritable traits and then find genes that way. But this is the way that people have been doing genetics for a long time. With the sequencing of the genome, a number of different technologies enabled reverse genetics, which is now that you know the sequence of those genes, you can either use, for example, an antisense oligonucleotide or RNAi, which was the technology of choice for about 10 or 15 years, where you can specifically code a double-stranded RNA, a short, I should say, 21 nucleotide double-stranded RNA that effectively incorporates into the cell's endogenous RNAi processing machinery, which in the end causes the degradation of a target RNA molecule. And so this, I should say, has been used for quite a long time, still actually has some uses, as I'll describe to you. But the key idea here is that you're introducing, whether by a chemically synthesized double-stranded short RNA molecule or an lentivirally encoded hairpin that in the end gives you that same product, a double-stranded RNA that causes degradation of its target. So now that we have the ability to knock down specific genes and the sequence of the genome, you can do genome scale reverse genetics, and that allows you to potentially find all the genes involved in your favorite process. So instead of just mutagenizing a plate of worms or bacteria and hoping that you find an interesting mutant that gives you the phenotype you'd like, you can really systematically scan through every possible gene, knock it out or knock it down, and ask what is the phenotype of that gene. And so I say that's easier to saturate because if you wanted to actually saturate the entire genome doing a chemical mutagenesis screen, you would need an enormous vat of worms or bacteria and thousands of people years to actually potentially get all the available mutants that might give you all the phenotypes. But if you know the sequence, you can actually go in and just surgically knock them down or out one at a time. So this is a much more efficient way of associating the gene with the phenotype, and now that we have not just the sequence of the genes, but actually all these regulatory elements in the genome that Luke was describing, we can actually approach systematic annotation of the genome, both for coding and non-coding areas. So there's a lot of information on this slide, but I want to just describe the two general strategies that have been used for doing genome scale screens. So on the right, you'll see arrayed library screening. This entire slide is geared toward CRISPR screening, but a very similar overall workflow was used for RNAi for a long time. And the key differences are that in this case, you have one well, one gene knockdown. And in this case, you have a pooled library where you have lentiviral, usually plasmids encoding either an shRNA or later a CRISPR guide RNA. And these are infected at low multiplicity so that each cell gets one virus in a pool. And then you can subject this pooled population to selection. And so in the end, in both cases, you end up with some selection strategy and then informatics to separate the hits from the non-hits in these types of screens. One of the nice things about an arrayed screen is that you can potentially get much more high-content imaging. So you can put these plates after you knock down one gene in a well into a microscope, take an image of what the organelles look like, for example, and really get an enormous amount of information for what's actually going on in that one well. You can't get typically that level of information in a pooled screen, but these have a number of advantages. They're much, much easier and cheaper to conduct. So I'll just tell you right now, everything I'm going to talk about from here on out is in pooled format because we love the pooled format. But I wanted to set this up because a lot of screens were done this way, and it's still actually possible to buy even now CRISPR guide arrayed in libraries because there are some applications where you really want an arrayed one gene at a time, one well at a time screen. Okay. So just to kind of go back to the history of these types of screens. So for a long time, screens were done RNAi screens were done in an arrayed format. And typically the workflow was sort of summarized in the slide before, but essentially you have a stock plate which has one siRNA in each well. You end up transfecting that into the corresponding well of cells through one or another strategy, and then you expose those plates to some kind of drug, for example. So in this study from Michael White's lab, they were looking at modifiers of a chemotherapeutic drug called paclitaxel, and they could find a number of genes, including proteasome, different microtubule kinetochore binding proteins, or modifiers of the toxic activity of this cancer drug. And so the utility of this approach was, can we find genetic modifiers that might be useful in the setting of cancer, where you use a drug like paclitaxel, which is frontline chemotherapeutic in breast cancers, for example. Sometimes that drug fails. Could you find another target that would actually improve the efficacy of that drug? So in some cases these work. Here's a pooled screen. Now each shRNA is encoded in a single lentivirus. These are infected into cells, and the screen here is to find, in the context of a KRAS mutant cancer, genes which, when you knock them down, further sensitize to another kinase inhibitor, a MEK inhibitor. So MEK inhibitors have been this powerful therapeutic that have been used in some cancers. They almost always fail because the cancer finds another way around it. And so this was a synthetic lethal screen to try to find combinations of second targets that would help a MEK inhibitor to kill KRAS mutant cancer cells, which in general are thought to be undruggable. So KRAS is this powerful oncogene that's mutated in a huge number of cancers. It's thought to be difficult or impossible to drug. So if you could find other targets that would work in a KRAS cancer, you might have a good way of killing these cells. And so in this pooled screen, they infect infected an entire genome-wide library, knocking down one gene at a time. They treated with this MEK inhibitor at a dose that would kill some of the cells, and then look for things that, when you knock them down, actually further allow killing of the MEK inhibitor. So this is what they found. The MEK inhibitor is called selumetinib, I think. And what they found is that in the presence of a control shRNA, the cancers still grow, but if you knock down BCLX long and you use this inhibitor together, now you have synergistic killing of these cancer cells. So this is to show that whether you're doing an arrayed RNAi screen or pooled RNAi screen, the technology actually works. I'll say that there have been enormous improvements in CRISPR screens from work from Luke and work from a number of other labs that I'll describe, where it's possible to really improve the specificity of these screens. Having said that, it's possible to do an RNAi screen and actually get useful results, and I'll describe a little bit more about this. So this was taken further, and they actually took tumors in mice and treated with that same combination of now an Avid drug that targets BCLX long and MEK inhibitor, and it could actually show tumor regression. So just to show that you can take an in vitro screen, use those findings to actually translate it to anti-tumor therapy. Okay, so while there were shining examples of that type in the literature where you could find something really useful, there were also these truly alarming examples of how terrible RNAi can be, and this is one of the reasons why people have been so excited about how dramatically improved CRISPR screens are. This is sort of a classic example in the literature of 3 different labs who all did an HIV screen. So ostensibly, this is a virtually identical screen. I believe they used the same cell type. It was a screen for resistance for HIV infection. I think they used different RNAi libraries. There were 3 different labs. But what you see here is that the overlap in their findings was actually probably lower than you would expect by pure chance, which is actually sort of impossible if you think about it. But it's one of those cautionary tales that is actually often cited in reviews as an example of how bad RNAi can be. And just to be completely fair to these folks, it's not like they're terrible labs that don't know what the hell they're doing. It's that there are fundamental problems that actually need to be considered, I think, in any screen, not just RNAi screens, but CRISPR screens as well. RNAi is subject to massive off-target effects that are typically not seen in most CRISPR screens, although I'll give some caveats for that in a minute. So Luke showed this amazing figure where you knock down with CRISPR-I GFP, and that's the only gene in the whole genome that gets changed. You would never ever see that in an RNAi screen, because if you put an RNAi species in a cell, of course, depending on how much shRNA or siRNA you express, hundreds of genes, in addition to the one you're intending to target, also change. So off-target effects are a major problem. With siRNA or shRNA, reagent heterogeneity is a major issue. It's still an issue with CRISPR guide RNAs, so we still don't know what is the perfect sequence of a guide RNA. But this is an even bigger issue with shRNAs. It's actually not that easy to find a good siRNA or an shRNA. At the same time, differences in methods for calling hits, cell lines, and the protocols that are done in different people's labs will always be a source of error, and so that should be kept in mind. I think even now, with the latest and greatest CRISPR libraries and people who are using what ostensibly should be 18cc-derived K562 cells, you sometimes only see 60% overlap between 2 labs that have done a nearly identical screen. So there are some factors that probably won't be solved in the near future that account for differences between labs. Okay, so enter CRISPR. So by comparison to an shRNA screen or RNAi screen where you're knocking down a transcription of an mRNA, with the initial CRISPR screens that were done, and of course this is now complemented with the CRISPR-I type screens